So welcome students again in our PYQ series. So today we'll be discussing about all the MCQs of biotechnology. So before going for all the MCQs in detail, you just revise the topic word to word. Got it? Make the points here very clear in your mind, all the concepts, all the diagrams, all the flow chart. Okay, prepare the mind map, right? It will be very helpful to you people to write to check out the correct answer. Okay, so let us start here with the biotechnology topic. So let us see question number 1. Preparation of curd, ghee, cheese and wine is based on the fermentation technology is called as. So of course you know that for the curd your mother is going to add 1 teaspoon of the curd in the milk, and next day you will have the formation of the curd. So this has been tradition. So we'll consider that preparation of the curd, then ghee, then cheese and all, right? So all these are coming under, that is nothing but we have the traditional biotechnology. Got it? You know that we are supposed to prepare the curd, then we take the butter, then we separate the buttermilk, then we are preparing ghee from that, but then we are having many more milk products here. So this has been the traditional thing here for all of us. So we can say that it's traditional biotechnology. Got it? So let us see which is the next question. Given below are the two statements. So students, let us read the two statements here in detail. Statement number 1. The recognition site of RENs, that is restriction endonucleases, okay, are 4 to 8 nucleotides long, characterized by the particular type of internal symmetry. Got it? Next is statement number 2. Different restriction enzymes found in different organisms recognize the different nucleotide sequences and cut the DNA at different sites. Got it? So let us see what are the correct statements here. Yes, this is the correct statement. This is also correct. So what should be your answer? Both these statement number 1 and number 2 are correct. So student, note down that A is your correct answer. Okay, read the statements carefully and remember the answer very perfectly. The manipulation of genetic material towards a desired end using in vitro, okay, process is called as. So what you will say here, it is nothing but genetic engineering. You know that cutting and pasting, cutting the desired gene and going when we put all these genes here into the new genome, so it will be the genetic engineering, right? C is the correct answer. Next, 4. Restriction enzymes actually cut the DNA at specific sites, that is dissolution of hydrogen bond between nitrogen bases, then cutting the nitrogen base at the 5th carbon atom of the sugar. Okay, next is cutting the phosphodiester backbone and separating the phosphate from the 3rd and the 5th carbon atom. So student, you know how the double helix structure is there of the DNA. DNA has got here, you know that this will be the ribose sugar. Got it? Here will be the nitrogen base. You know this very well, which is going to form the bond between the another one. Okay, so you know how the sugar molecule is going to be there, right? So here will be the phosphate group, here will be the another sugar, okay, then here will be the phosphate group, here will be the nitrogen base, right? And your enzyme is going to cut the sugar phosphate backbone. Got it? So this will be the correct answer, cutting the phosphodiester backbone, right? Then you are having, this will be the right action, okay, of what here? Restriction enzymes. Restriction enzyme is going to cut this bond, is going to cut this bond at the specific site, okay? So what should be your answer here? That is okay. Next, 5th. The bacteria protects its own DNA from nucleolytic attack of nucleases by dash methyling all the bases of the DNA, then to help the alkaline phosphatases, then methylating the bases at susceptible sites, and help the DNA help of DNA ligase here. So obviously there will be methylation of the base here at the susceptible site. So students, please mention this very perfectly in your notebook that methylation is going to be there. Okay, so C is the correct answer. Next, a very good vector which is used in recombinant DNA technology should have the following characteristics except, okay, which is not going to be there. Number one option, ability of the independent replication, must contain unique cleavage site on one marker gene for the restriction enzyme, should have at least suitable control elements like promoter and operator, ribosomal binding site, and should have the repressor mRNA. So what should be your correct answer? Your correct answer here is D. D is your correct answer. Should have the repressor mRNA. Okay, so D is the correct answer. I'm sorry. Next is which of the following, okay, is considered as molecular scissor in the field of modern biotechnology? Let us see what is that. Answer option number one, reverse transcriptase, Taq polymerase, all alkaline phosphatases, and restriction endonucleases. Obviously, REN, that is restriction endonucleases, will cut the bond at specific sites. Got it? So it will be the what here? Molecular scissor. Got it? Molecular scissor is restriction endonucleases. Okay, now the next is very important here. So what is that? Let us see. See column number 1 and column number 2. Column number 1 is giving you LU1, BAM-HI, EcoRI, HIND2. Okay, and they have given the sites here. So what should be your correct answer here? A is the correct answer. Let us match the following. Number 1 is A, number 2 is D, okay, number 3 is C, and HIND2 is here B. Got it? So A is your correct answer. Student, in your textbook, okay, we have a very good chart which gives you all the details here. So please go through this chart, okay, in detail, right? Next, read this statement. Plasmid, that is PACYC177 is one of the constructed plasmid, okay, and plasmid PBr322 is mostly used. Of course, yes, it is used here mostly, frequently in biotechnology. So we have both these statements are correct. This is also correct, this is also correct, because both these are very useful here in the field of biotechnology. Next, what is the time duration required for completion of one PCR cycle? You know that PCR is what? Polymerase chain reaction. You know the steps in this reaction. So please go through the diagram. This diagram will give you denaturation, then it will give you the annealing process, and it will give you the extension, and then you are getting the daughter DNA which is formed, right? So how much is the duration? How much is the time required for that? So it is around 3 to 4 minutes. Okay, 3 to 4 minutes will be the ideal duration for the cycle. Next, which of the following is used in vitro amplification of the gene? Of course, you are using PCR, polymerase chain reaction. You know, you are using Taq polymerase, which is acquired from the Thermophilus aquaticus, which is the bacteria used here, Thermophilus aquaticus. Okay, please remember, okay, Thermophilus aquaticus. It has got the, we say that it is nothing but what here? Taq polymerase. Taq polymerase enzyme is isolated from this bacteria which can tolerate the high temperature above 90 degree. Next, question number 12. Elimination of the defective gene and insertion of healthy gene into a genome is achieved by dash. Of course, we are using here, that is recombinant DNA technology. You know, cut one gene and paste it. Got it? Cut, paste. Okay, you are cutting this gene. Okay, if you have got this gene and you are going to put this into the vector, right? So this is going to be very unique here in the recombinant DNA technology. Read the statements, student. Number 1, during PCR, in an automated thermal cycle, denaturation, annealing, and primer extension, all these steps are automatically repeated here 20 to 30 times. At the end of the N cycle, the PCR 2 raised to N copies of this. Okay, so copies of the segments are produced here. So what should be your answer? Your answer here, statement number 1 is correct and statement number 2 is incorrect. Correct, incorrect, and your correct answer here is C. C is the correct answer. Next, which of the following statements are correct? Exonucleases cut the nucleotides at the ends of the DNA strands. Okay, read the statements correctly. Sequence recognized by restriction enzymes 4 to 8 nucleotides long. Baccalovirus are not vectors. And what is next here? Agrobacterium tunificiens is a bacterium which can live only in plants, and bacteria protect their DNA from nuclease attack by methylating the susceptible site here. So what should be your correct answer? Your correct answer here is D. These are the only correct one. Number 1, number 2, and number 5. Got it? So these are the correct ones. So this is the correct answer here. Next, in vitro amplification of DNA in PCR also requires appropriate quantities of course, you need to have magnesium. Okay? Remember this. Next. 16. DNA segments with dash number of nucleotides will move fastest towards the anode during the electrophoresis. So it is about 5,000. It is about how much here? 5,000. Okay. So please remember this answer. Next, what will happen? The cloning vector does not have ORI. What is ORI? Origin. You know that during the process of replication, you have one point which is giving you the starting for the replication is ORI gene. Okay. So what is that? It is the replication of host DNA only. Okay. Next. Question number 18. The technique of combining the DNA of two organisms into a single gene is called as, so we will obviously call it as genetic recombination. Very, very simple. When two genes, when you are having the crossing over, in that also you can get the genetic recombination. Next, the cloning organism used in plant biotechnology are, okay, E. coli and Agrobacterium tunificiens, E. coli and Bacillus thuringiensis, Bacillus haemophilus and Helicobacter pylori, E. coli and Bacillus haemophilus. Okay, so what should be your answer? Obviously, you are having along with the E. coli with Agrobacterium tunificiens. This is very, very common and very frequently used bacteria. Okay, A is the correct answer. The given restriction site is cleaved by which one of the following restriction endonucleases? Obviously, this is nothing but HindII. Okay, remember, this is the site, HindII. Next. Which one of the following is not required in PCR? Magnesium ion heat stable DNA polymerase, RNA polymerase enzyme, ribonucleoside phosphate. So obviously you are not having the use of this. Okay, so C is the correct answer, that is RNA polymerase enzyme, not required in PCR. Okay, next. Plasmids are the ideal vectors gene cloning because they have the ability of, they have independent replication in competent host cell, replication freely outside the bacterial cell, multiplication in the culture medium, carrying very large DNA inserted into the host cell. So what should be your correct answer? Your correct answer here is, that is, we have independent replication, we have independent, so we have, this is having independent replication in competent host cell. A is the correct answer. Next, denaturation in the is the correct sequence steps involved in PCR. Obviously, what should be there? Denaturation, annealing and primer extension. D is the correct answer. So please remember this. Once you are having the denaturation here, okay, then we have the attachment of what here? Primer. You know that primer is attached here. Okay, here you have the attachment of primer. Okay, this will be your primer. And third one is nothing but what here? Extension, right? Third will be what here? Extension of this. Okay, so you will have the extension of all the points over here. Extension will be here like this. Got it? So these are the steps here. Next, question number 24. The technique of PCR was discovered in 1985 by, of course, Kary-Mullis, right? Kary-Mullis is the scientist who discovered this PCR. Okay, next. Question number 25. The transfer of recombinant DNA into a bacterial cell for the purpose of its cloning is assisted by, of course, it is assisted by calcium ions. You should know that calcium ions are very important here in this process. Okay, so these are the questions here. So please go through all the details.